To emphasize what happens to the DNA during the cell cycle, we are going to be making a little booklet. You will need 2 different colors of pen, pencil, marker, highlighter, something similar. The first step of the cell cycle is interphase, which consists of 3 subphases, G1, S1, and G2. During G1, the cell just lives and grows. The DNA exists as chromatin, which is uncoiled DNA in the nucleus. Take one color and draw some spaghetti-looking DNA, and only one color because right now there's only one copy of the DNA. During the synthesis phase of interphase, the DNA is copied. It has to be uncoiled to actually make a copy of it. So go ahead and draw with 2 different colors showing the uncoiled DNA, the original, plus the copy. During the G2 phase, the cell continues to grow and also does some last-minute preparation for mitosis. For example, it makes extra centrioles, extra mitochondria, extra Golgi apparatus, lots of extra organelles in preparation for splitting them between the 2 new cells. The next step in the cycle is mitosis. Mitosis splits up the DNA in the nucleus. Cytokinesis splits up the rest of the cell in the cytoplasm. There are 4 steps to mitosis. PMAT, prophase, metaphase, anaphase, telophase. Prophase is preparing to split. The DNA coils up into chromosomes. So it was chromatin, and now it's chromosomes because it's coiled up. Notice there are 2 sister chromatids in each chromosome, one with the original copy and one with the identical new copy. So this cell happens to have 4 chromosomes, 4 pairs of sister chromatids. The nuclear envelope or nuclear membrane dissolves. You notice there's no more nucleus. And the centrioles start to form spindle fibers. So draw some lines from the centrioles showing those spindle fibers and draw 4 pairs of sister chromatids, making sure to use 2 different colors to show the original copy and the new copy. During metaphase, the chromosomes line up in the middle of the cell, middle. And spindle fibers attach to the chromosomes at the centromeres. So there's the centromere, where the chromatids are attached to each other into a chromosome. And you'll see here's the centrioles making spindle fibers that attach to the centromere of each chromosome. The chromosomes are lined up in the middle of the cell. During anaphase, the spindle fibers pull the sister chromatids apart, anaphase apart, into the individual chromatids and individual chromosomes. So each of these chromatids is pulled away from the others. Notice how they were kind of turned like this. Now they're kind of turned inside out because they're being pulled away from the center by the spindle fibers. Next is telophase. It splits in 2, telophase 2. The nuclear membrane begins to reform, and the chromosomes are still coiled up. They're getting ready to uncoil, but they haven't uncoiled yet. And you'll notice that the cell is also beginning to split. At the same time as telophase, the cell also splits in 2 through cytokinesis. So mitosis, telophase being part of it, splits the nucleus and the chromosomes. Cytokinesis means cyto, cell, kinesis, cutting. That cuts the rest of the cell with the cytoplasm and the rest of the organelles. In an animal cell, cytokinesis occurs as a cleavage furrow forms. Yes, it's called cleavage, and Yes, it looks a little bit like it. And what happens is it pinches the cell and pinches it in half. And where it pinches is called a cleavage furrow. A furrow is like where you dig in the dirt and make a furrow or a ditch in a field and then plant the seeds in the little ditch or the little furrow. So a cleavage furrow is cleaving the cell in half, but you'll never forget it because, well, yes, it looks kind of like cleavage. If there is a plant cell, then what will happen is a cell plate will begin to form in the middle, and the cell plate will turn into a cell wall. Finally, each new daughter cell returns to interphase. So the DNA uncoils, the nuclear membrane is completely formed, and the cells are totally separate. These cells are identical with identical copies of DNA. Each cell then continues on in the cell cycle until each one is ready to undergo mitosis.